I can spend a little bit of time while people join, just introduce myself. As Tony said, most of you do know me, and I'm delighted to see some people on the call. Haven't seen for a long time. My background is I've been a virologist for about 40 years, in academia, then the Wellcome laboratories down in Beckenham and Kent, then made my way up to Stevenage, joining GSK, head of antiviral research there, and then left to join a strange Swedish biotech company called Medivir, where we developed a hepatitis C drug, which was marketed by Janssen and went into several 100,000 patients, which I'm very proud of. And then joined Viral about 4 years ago. And we're working exclusively on RSV, but I'm not going to talk about Viral today. So Tony, do you want me to, Graham, do you want me to launch right in now?
Yeah, hit it.
Okay. Yeah, that'd be good.
So let's just go for the first slide. This is hopefully this is going to be a fun 2 or 3 slides, but also provocative. The provocation comes towards the end. So pandemics, past, present, and future. There's been a long history. People have known that viruses and other infectious diseases go from animals to humans. Very well established. A particular case I actually like is a bacterial one, which is plague, which seems to have started from a gastrointestinal disease in humans that occasionally went into fleas, but not very well, because one of the plague genes, urease, actually killed the fleas. So the first mutation, a single point mutation happened, which inactivated the bacterial urease. And now the fleas could survive being infected by the bacteria. And that meant they got some local transmission into humans. And then there was a second single base pair change, which meant that the clotting agent that the fleas use when they bit the human was more effective. And so the bacteria they carried got more deeply penetrated into the tissue. And that's when we got a sort of epidemic of plague, 2 single base pair changes. Smallpox is another virus that's thought to have come from an animal a long time ago. We're all very aware of influenza virus, which is actually a virus of birds, okay? And it very often goes from birds, sometimes to an intermediate, often the pig, and you get a genetic reassortment in this process that gives rise to a whole new strain of flu, which causes pandemics. And we've all seen the H1N1 pandemics, etc., and Spanish flu many years ago. The next one is HIV, which emerged in the sort of 80s from various monkey species into humans. Ebola comes from bats. Rabies comes from most animals that carry rabies. And last on our list, but it could go on for a lot longer than this, is our favorite topic of the month, coronaviruses. So zoonotic infections are not new. Let's look at the next slide. So this slide's going to tell you a little bit about coronaviruses as a family. It's a big family of viruses, okay? I've pulled out 3, SARS, MERS, and COVID-19 in the order that they transmitted to humans. So the first thing you can say, in the last 16 years, 3 coronaviruses have gone from bats via an intermediate vector into humans. So this virus group is pandemic ready, okay? There's about 30 species of bats already. There's probably a lot more. And these group, they have to have one single change to get them into humans. They normally change about 30% of their RNA, and they introduce the capability of infecting via this receptor, the ACE2 receptor. Once that's achieved, there are some other single point mutations that make this infection more effective, but effectively that ACE2 change allows the viruses to get into humans. And so far this has happened at least 3 times, possibly more. So if you look at the table and you go from SARS to MERS to COVID, you move to the right, you see lethality. And SARS was about 10% lethal. Now, I'm introducing a concept here called R0. That's basically simply explained as if you're in a room with a couple of 100 people and they're all susceptible to infection, then R0 of 3 says you infect 3 people in that room. Generally speaking, an R0 of less than 1 is unlikely to transmit very effectively, so you're not likely to get epidemics. An R0 greater than 1 and that becomes increasingly possible. Give you an example, the R0 of measles is about 18, and measles is a really damn effective infective virus. So SARS, estimating now the R0 was between 0.1 and 1, and that may explain why this wasn't a particularly infectious virus. And it was contained largely in China with a couple of outbreaks in the likes of Canada, but was contained by quarantine. Then we have MERS. MERS is a real worrying virus. 35% lethality. Yeah? 1 in 3 people catch MERS die. It's actually still transmitted in the Middle East, but look at the R0, it's about 0.3. So basically, in the Middle East, when you get some of this virus, you lock them up and you lock all the families up, and basically that contains it. And then we have COVID-19. It's a bit early to start speculating, but I will do anyway. Lethality is probably about 0.5% to 1%. That's what we're going to find, except this will vary an awful lot according to the population that the virus is spreading in. So we know that in Italy, for example, northern Italy, there are a high degree of elderly people who happen to live with a lot of younger people. That's a really bad combination, and that's probably why they've got a very high incidence of a mortality rate. Look at the R0, again, it's a bit speculative, somewhere between 1.5 and 3.5. So this is a virus that can, and that's what we're seeing now, cause and spread and cause epidemics. Okay, so let's move over to the right. All these viruses, SARS, MERS, and COVID, all came from bats. I said before, there's 30 known coronaviruses in bats. They went via a vector. We know in the case of SARS, it was a cat, a civet. A little picture of a civet there. We know that MERS, ironically, came from a camel. And in COVID-19, the speculation is it came from a pangolin. That nice looking little animal there that in China, they like the meat, it's a delicacy, and they make Chinese medicine out of the scales on the animal. So that's the background. Now, one of the themes I'm going to develop in the next slide is if you look at those lethality rates and those R0s, we have dodged a bullet here, even with COVID causing the disruption it has. We're basically being very, very lucky in how this virus spreads an infection. So the next slide talks about potential approaches. Whenever a new virus emerges, that's hepatitis C, Ebola, SARS, MERS, a whole load of treatments emerge. Usually there are totally unrealistic timelines given to development of these treatments, whether or not they're small molecules or vaccines, and an underestimation of the safety and the development timelines. There are usually a whole load of open label clinical studies done, small numbers of patients, not controlled properly, very biased groups, selection groups, and basically most of this data that's been generated in all the viruses I've talked about is usually completely useless. And I would put it to you that the clinical data that's been commented on to date in various countries, I've read it in great detail. I encourage you to actually look at those papers. But actually, I feel they don't tell me anything whether or not this is going to work or not going to work. These areas, these viruses, become heavily funded by pharma and NGOs and governments. SARS was 2005. You want to get a grant on SARS, it was dead easy. There was loads of money going around. 2 years later, you couldn't find money to support SARS work because everyone said, it's done, it's gone. No need to fund it anymore. And I think we've got to be very, very careful as a society to take the bottom bullet point. We've been extremely lucky. We've had a warning shot. We need to plan and work. Yes, we need to approach COVID-19 as a high priority, but we also need to have our eye on the ball for COVID. And I've given a number here, COVID 2025, but it might be COVID 30. There will be one in future. Yeah, N equals 3. In 16 years, we've had 3. And if the mortality was that of MERS, 35%, and the R0 was 4, we'd have up to 1/3 of the world's population dying. Yeah, that is not impossible, those combinations. And so this, I think, however horrible it is, we need to take as a warning shot to get our act together and make sure that money does not disappear once COVID-19 starts to ease off and disappear into the background. Money and funding must continue to be funded. Now, the last bit before I open this up to everyone is to tell you what we're doing about it. So Reviral, the company I work for currently, is not doing very much. We're entirely focused on another virus called respiratory syncytial virus, which kills a mere 200,000 people every year, babies and adults. And we've got a very tight focus. But some of us who work together very closely are about to fund or set up a non-for-profit.
Sorry.
Trust it'd be you, Tony, for goodness sake.
Yeah, there we go.
Every time, actually. It's very interesting.
You spoiled my punchline, Tony. Let me carry on again.
Carry on again. Sorry, Eddie.
We're going to set up a not-for-profit organisation. In fact, they'll get set up next week. We've already raised, last Friday, we raised £100,000 to fund this. And our first 20,000 compounds are actually on the way to a screening group over in Belgium from a big pharma. And these are a set of compounds which the viral scientists developed in previous companies, which were targeted at viral RNA polymerases, so hepatitis C and RSV. And they're already winging away to a group, an academic group in Belgium who can screen these very high throughput against COVID-19. Our feeling is that we need to, of course, focus on COVID-19. However, we also need to stand back and say, we need molecules in place, ready to roll, that can hit the next coronavirus that comes through. And there's a particular profile we want to generate, which I won't bore you with today. But we have those plans in place, and screening should start in 2 weeks' time. Okay.
So, Eddie, Fab, man, can we take your screen off and bring all the lovely people's faces on?
Oh, of course we can. There we go.
On the,
Stop share. There we go.
Man, so, you know, Fab, double Fab. I mean, the essence of these coffees is that we stir the pot and we get people talking. So, you know, this is it is not meant to be a sort of, how do I describe it, scientific thing. But I think Eddie's provoked us a lot. And, you know, when I spoke to Eddie earlier, and I suppose part of me said, should we shoot all the bats? Which was, you know, my initial reaction. Second reaction was a bit like, well, you know, why are we talking about it? What's all this about? And, you know, but there's clearly a benefit for us all as scientists, or once upon a time scientists, to do it in our head. So I'll ask people to ask questions, and if you don't ask a question, I'll pick on you. So someone better ask a question.
I'll ask a question.
John, man.
So Eddie, I should have the same background as you, but not quite. You went on a bit more professionally than I did. R0, to what extent is it a function of the virus, and to what extent is it a function of the various hosts that it comes into, that the virus comes into?
Yeah, good question. Thanks, John. It's good to see you again.
Basic question.
So I think the policies, let me answer a little bit differently. I think the strategies that most governments are trying to do, with the exception of one or two more maverick societies, is effectively to control R0. So we shouldn't see R0 as an immovable object. I think it's the first thing. You can measure it in a purest way, as the model I used before. But if you can prevent, you can, by social isolation, you can take that room I talked about with 100 people in, you can take that down to 5, then you're starting to reduce the R0 right away. I'd also argue that the approach that myself and Ken Powell and Stuart Cockrell are coming up with to develop new coronavirus compounds, which are not designed to be very potent. Once you get potency, you tend to lose broad spectrum activity. So what we're looking for are more broad activity with very good PK and very good safety that can be stockpiled and used immediately as new coronavirus. They may not be potent enough to eliminate the virus right away, but they would provide what I would call chemical chemical quarantine. They provide another way of reducing the R0 and giving people time to plan and to activate other measures that can be used. So it is, John, it's based, R0 should not be seen, in my view, as some sort of purest measurement. It should be seen as a number that we can change and modify to control the disease.
So just one question, Eddie, before I find, just stirring the pot a bit more. I mean, if you were to take a photograph over GB or the greater GB, how many people are working on this stuff? You know, you've talked about, you know, where are the repositories of, you know, have we got 50 people doing epidemiology? Where is this centered knowledge? Is it?
It's emerged very recently. The group that's been dedicated into coronavirus in the UK for a long time is Jonathan Ball's lab up in Nottingham. Now, he's worked on coronavirus scientifically for a long, long time. And the group, I think, in the UK that gives the most credible epidemiological information is a group in Imperial. Now, there are other groups, and groups are working on it increasingly day by day. But I think if you want to look back at someone who really has stayed in this area for a long time, and I like those people, because they basically tend to increase the knowledge base, it's Nottingham and it's Imperial.
Yeah. Okay, question from all. Panteli, you ask a question. I can see you there. You're under my icon thing. Panteli.
Yeah, no, thanks, Tony, and Eddie. Thanks for the for the presentation and the fabulous insights. I'm one of the global medical leads at PPD, so a physician by training and an immunologist by training, actually, with a focus on cell and gene therapy. One of the questions I was interested in kind of asking you is, what about the development of sort of immunological profile and reconstitution immunity in these patients? You know, what's your thinking there? Because we hear about, you know, these biological phenomena in patients. What are your thoughts in terms of that immunity and long-term immunity in that?
Okay, I'm really glad you asked that question, because it's a very important one. I had to put my hands up first of all and confess I'm not very good at immunology. It's sort of, I was trained too early to pick up the more recent developments. I also confess my youngest daughter is an immunologist, so she tries to teach me every so often, but it's a foregone. I can't do it. Anyway, I'll tell you what I know. I think you're quite right to say that immune pathology of COVID infections is critical. Now, what I want to do is draw you upon the virus that I've been working with for the last 4 or 5 years, respiratory syncytial virus, and some very appropriate lessons. So the first one is RSV, I said before, kills 200,000 people worldwide. About 14,000 old people a year die in the UK. Yeah, about 1,000 babies. So it's a very important clinical need and commercial opportunity. People have been trying to develop vaccines against RSV for a long, long time. The first one was in the late 60s. It was tried in the States. What happened was the babies that were not vaccinated, the control group, were fine. They got sort of normal RSV when they got it. The babies that vaccinated were nearly all hospitalized, and some of them died. And they died because we generated an inappropriate immune response. Second thing I'd say is, after a few years of blind panic, no one will go near RSV vaccines for several years. And then eventually people worked out what the problem was and came up with the next generation vaccines. And since then, there must have been 30 different vaccines: purified protein, messenger RNA, recombinant pox viruses, you name it, everything's been tried. They nearly all work in phase 1, phase 2. So in other words, you can elicit an immune response to the RSV protein. That's not rocket science, is it really? You know, you'd know as an immunologist better than I would. You stick an alien protein into someone, you're going to get an immune response to it. None of them to date have given any protection in any patient groups. And so again, and if you look at RSV, you can get 2 or 3 infections before you get full immune protection. So I'm a little bit concerned with some of these vaccines, which are just going right into humans without any animal model data. There is some animal model data that came out the last couple of weeks, which clearly shows in primates that you can elicit, if you immunize them first, get a high antibody response to S protein, which is what most people are trying to develop, is this spike protein that binds to ACE2. if you then infect controls or the vaccinated animals die. And that gives me the shivers. I think we've got to be very, very careful how we immunize people. But to come on to your broader question, I'm very encouraged to see some of the clinical studies using IL-6. I think that is coming up time after time as being a major problem. I would also, I'll give credit where it's due. My younger daughter keeps on poking me saying TNF alpha, TNF alpha, TNF alpha, and she's going to phone me tonight and tell me why she says that. Third attempt to explain it.
Just one stir in the pot. I mean, when we talked earlier, you said you got a glimpse when old Patrick said that about herd immunity. Eddie, what was your glimpse to what he was saying?
I think what he said was probably perfectly true, but it's quite, people don't want to hear it. I think before we get an effective vaccine for the general population, we may get one for the healthcare workers before then, and I hope we do, or before we get a small molecule that really works effectively. I think by that stage we've all been infected. And I think once we get infected by, let's say, Christmas time, and we have 30, 40, 50% of us infected, that R0, to go back to John's question, starts to drop. Now, herd immunity is normally above 90%. That's what people like to see with a vaccine to show that you stop the spread in the population. But I think we're going to see benefit from even having 50, 60% who have been infected and recovered. And the other thing I point out to you is that 80% of people have a mild infection of this virus. About 20% have no symptoms at all. So I think there's an element of trying to control the spread, and I'll call it a controlled burn, protect the at-risk populations, keep them out of it for the next 3, 4, 5, 6 months, and then basically hope that herd immunity starts to increase to stop the spread later on. I think Patrick probably just, unfortunately, went a bit too far on exposing the thinking of the powers that be. That's my guess. I think it will happen.
Let's ask a question. Tom, a classical pharmacologist view.
Yeah, well, this classical pharmacologist, his first job was working on smallpox.
Oh, wow.
And infectious bronchitis at Evans Medical many years ago.
Yeah.
Yeah, so he has some appreciation, and microbiology was his first degree. So bacteria, viruses, and all those things I've had a great interest over many years. To a comment and a question, Eddie. The comments, just listening to CNN earlier, they were saying this virus is more like measles in its infectiousness. So, you know, where this came from, I don't know, but basically it's spreading as quickly as measles does. So that is a comment which you can, perhaps talk about later. But one of my sort of interests as a pharmacologist is the way this sialic acid is becoming known as a co-receptor for the ACE receptor, and that you can knock out in certain glycosylation states. Yeah, you can knock out, these S or N glycosylation states. And, ways of doing that obviously are, small molecules, pharmacological, etc. But the siRNA technology seems to be one which I don't know whether that could be more rapidly advanced than, say, the antibodies. So there's a couple of things to think about and comment on.
Sure, thanks. I hadn't heard the CNN report. I'm surprised to hear it because I keep on banging on about this R0, but the R0 for measles is 18. It's highly infectious.
Yeah.
I frankly don't believe this is going to be as high, but there may be other common features. But I hadn't heard it, so I'll have a read and see what I can do. Your second question is really interesting because when there's some data came out today showing that COVID-19 has actually had a deletion in the site that's responsible for the glycosylation of S protein. And it actually prevents it from being cleaved by some of the host proteases. And there's speculation because it's all speculation at this stage. Most of the science probably is all speculation, but the speculation that's increasing is pathogenicity by that single mutation. Remember I said at the beginning there were over mutations. So there's that mutation, which is really interesting. There's a couple of mutations that make the virus translate its proteins more effectively than does SARS, for example. So there's obviously some tweaks that have gone on here. But I think you're right. I think there are other sites. The targets for coronavirus, as I see now, the dyed-on-the-wall classical virologist one, which I'm one, would be RNA polymerase, top of the list. It tends to be conserved. We've got lots of inhibitors of viral polymerases in HIV and herpes and Hep C. They work. And we've got libraries against those that work. So polymerase would be the top of my list. Next will be the protease, so-called 3C protease. The SARS has a crystal structure. Diamond did a great job in the SARS protease. All the information's out there. If you want to sit down and design yourself an inhibitor, it's all there. And it's obviously involved in processing the viral polyprotein, and that's essential. If you don't chop that polyprotein up, nothing's going to work. The last one is the S protein, and ACE, and I think it's a credible target as well. Probably harder to develop, but I think it also should be tackled. But then you're talking about other targets, such as the glycosylation pathway.
So
and again, I think they'd be second tier, but I would look at them.
Yeah.
So
Thanks, Eddie. Thank you.
just sort of, one of the things, we're a life science community, a melting pot of people. So maybe I can ask somebody who's not like some of us getting the old manuals out and checking out on our knowledge of virology and things. So maybe somebody who's in one of the other parts of the life science community, either Sue or Pilar, I see. These are people who are maybe not virologists, but questions to Eddie from the non-virologists.
Denise as well.
Denise, yeah.
Yeah, okay, I'll ask a question. Hi, Eddie. It's a long time since I've seen you.
Yes.
I'm particularly interested in the antibody response. My husband, Professor Case Lucas, is hidden away in rural France because he's got a lot of comorbidities. And we've decided we're not going to meet until I've actually had it and can prove I've got the antibodies. So I'm curious to know about any thoughts around reinfection. You know, there's the whole thing about the antibody test, but we don't seem to have any thoughts yet about whether that's a valid protection for any length of time. Any thoughts on that?
Yeah. Okay. So if you remember what I was saying earlier on with RSV, that virus has to have up to 2 or 3 infections. So babies get it. They don't always, but they can get it again, sometimes up to 3 times before they then get lifelong protection. If you look at the elderly, when they undergo immune senescence, again, the immune system tends to decline, and then they get back into RSV infections, and they can have 2 or 3 infections in a season, again, boosting the immune response. So that's, I think, the worrying sign, that maybe you might need to have several infections. However, I find it, without any data to support me, but that's never stopped me before, I find it difficult to believe that you will not get a protective immune response. But it may be there is a certain fraction that may be quite large who will need 2 or 3 infections to get that immune response. Other coronaviruses, there are a natural group of human coronaviruses which have been established in the population for a long time, and they tend to sometimes need 2 or 3 infections to provide immunity. So there's a worrying sign there.
So just one point, as we try and get more people involved. I mean, it looks like the news wires, and I'd be interested in John's perception on this, is that almost every day it's a bit like there is a particular topic for a day. So I think today's topic is masks. I think yesterday's topic was testing. And I just wonder, I've got this resonating in my head, John, that you said when we had our talk earlier in the week, that if it wasn't for the news, we might not even notice this. I mean, so one of the questions I think is about, it's just the way our brains are being reacted to all of this stuff, and whether, I don't know, whether we're a substrate for some sort of anxiety thing, which is, you know, I don't know whether testing actually is irrelevant. I'm not convinced it's totally relevant. But if we go around it, you know, thoughts about all of that, and anyone who wants to say anything, and ask questions to Eddie, of course.
Hi, I actually have a question linked to Tony. Good afternoon, everybody.
Hey,
Eddie,
where are you, Pila? Where are you?
I'm in London.
In London. What's London like?
You don't notice anything unless you are in hospital, of course. And this is my question to you. I mean, from a population health perspective and management, from what I hear, we're not here for the next 6 months, we're here for the longer term. So my question to you, to Eddie or anybody who like to chip in, if you were given, you know, the command on how you would manage this pandemic, what would be the things that would be asked from the public, from the business, in order, you know, to manage that? Because obviously, like, there is an immediate emergency now, but people still need to go eventually to work and live on something. So how would you manage that? I mean, it's like that we are not doing already.
I'll have a crack at that. Much as it really peeves me to say this, because I'm not the biggest supporter on the planet of Boris, I actually think the strategy we have is not being too bad. You know, we're trying to achieve a balance between locking up the very at-risk patients while frankly allowing some of the more healthy people to get out there and get the damn virus. I think that's not a bad strategy. The big question we don't know that really worries me is whether or not this virus will be seasonal. Whether or not it's going to make, again, I'll go back to RSV. You know, RSV now comes into the UK, you see RSV cases around about November to around about March time. And then it goes down from the Northern Hemisphere to the Southern Hemisphere, and places like Australia, New Zealand. New Zealand gets it around about May, get it for the summer. In the tropics, they tend to get like this grumbling infection that goes on for most of the year. Now, the problem is, are we about to have a good period of time when we don't see it in June, July, and August so much? I don't think it's going to be zero, but it's going to reduce. And then we're going to get a second wave in the winter. Now, to a certain extent, I think that's what the government are trying to do. They're trying to keep it down for now, hope to hell that the summer reduces the natural transmission, and then prepare better with ventilators and hospitals and just the general infrastructure, which they dismantled, of course, get that ready for the following boom, because it may come back over the winter. What else would I do? I think the big problem we've got now is testing. And Tony, you said this, but it was yesterday's story. I think it should be the story every day. We are not testing anywhere near enough people. Nowhere near enough.
What? Just answer me this, Eddie. What question are you asking with the testing? Two.
I want to know who has it, so that we can put them and their close relatives definitely into quarantine. We're locking up too many nurses and doctors on the presumption that someone they're close to has this, and they may not do. And you know, about 25% of medical staff are in quarantine now, maybe inappropriately, and I think that's unfortunate. The second one is I'd want to know who has seroconverted, who's generated IgG, and I would issue those people with a little passport that basically says I had IgG detected in June, and it's now November. I'm pretty safe to move around the place. And I think
so one of those is tactical.
Yeah.
So the first one is tactical. How do you save how do you sort your workforce out, or your resources? And the second one is a measure of safety, almost, isn't it? That you've got people out there that can move around that theoretically are safe.
And one of the questions from Pilar was around how do we get to normality? I think I'm abstracting it. I think that's, and one of the ways we can do it is by using these two tests appropriately, only assisting people quarantine when they've had a positive test to show they've got the virus, and then allowing people who've had it and recovered to start moving freely. We can start to get some percentage of our population back into circulation. I think that's really critical. I haven't seen any strategy from our government about how we're going to end this process.
No, that's exactly right. Other people would. So let's hear some other interesting people.
Can I just follow up on that on the testing? Eddie, would you like to stagger the testing so that you get a time sequence for spread of infectiousness?
Yes, but I think we've got to be pragmatic. I think now we need to target populations. If you look at what South Korea did, they did a fantastic job. We can't do this in our society. We have to accept we can't do that. We don't. British are damn awkward. We're not going to do what the South Koreans did. But on the other hand, if you can screen, and the Germans are doing a great job at screening as well. I would focus on the healthcare workers. You know, make sure they're covered and clean and safe, and get them back in where they're able to do so.
I mean,
once you've done that, then build out of that population.
I sense there's a big feeling of masks being used. It feels like the next announcement's going to be masks.
Yeah, I won't be wearing one. They look stupid.
I've got my own World War II gas mask out. Mr. Producer, have we got, Mr. Producer, who's new on our?
We have a Mark Withers on the line who has just joined us. So
Mark,
yeah, it'd be great to hear from
tell
you.
us who you are and where you are, man.
Are you on mute, Mark?
Oh, he's off mute now.
Unmuted. So hi everyone. So I'm Mark.
Hi, Mark.
I'm coming in from Abingdon. So I'm not a scientist. I work in the field of organisational development. So I was really interested in what you had to say, Eddie, about this sort of ballooning of investment and activity, and then the sort of disinterest once things start to get back to a degree of normality. What I'm interested there really is how do you resource, scale, get the right people working on stuff that's actually going to be sustainable and has some degree of long-term impact rather than this salience of where we have periods of energy and then disinterest. I don't know if that makes sense. That's
yeah, no, it does. It does. I don't have a great answer for you, but I'm going to give you the negative side. I don't think we can expect pharma to do this. They're driven, understandably so. They're there for the benefit of the shareholders. They're there to generate profits. And if they can't see a profit being made in 5 or 10 years' time, they're not going to do a lot of investment in that. Now, having said that, there are some exceptions. My old company, GSK, has invested in TB and malaria in various ways using NGO and government support to offset the costs, and done a very effective job. So they can do that, but they need to be given a framework which encourages them to do it outside of the profit side, because they're never going to get in it to make money. They know it's not going to make money.
Because sorry, can I just say, that sort of COVID 2025 that you painted, the spectre of that, I mean, that would reinforce what you just said, I think, Eddie, that there needs to be some degree of plan and that this isn't going to be a one.
I mean, some of us are old enough to remember government scientific institutes, which my favourite was agricultural research. I worked there, and it was like a big city of people work for the government. Now that's gone. Now there is no. Is there much institute? You know, there is not much government.
The Wellcome Trust. Patrick Valance is working closely with the strategic group at the Wellcome Trust. And I think that's a sort of vehicle to look at for the future with regard to, you know, bringing other constituent people into that sort of, you know, strategic organization. And, you know, that then could link up with, you know, the American organizations, etc.
Yeah, I agree with you. In fact, there's a good precedent for that, which is the Wellcome Trust on Carbex for antibacterials. It's done exactly what you say, linking US and in UK.
Also, for COVID, there is an initiative launched by the BIA, where everybody's there, the Crown Services and all the scientific community. I can share the document because they're there to act as an umbrella for all the UK. I believe all contracts and everybody's involved. So
just
another thing is, sorry?
just to help us, what's the take home from the BIA? What are they?
The BIA basically is acting as a coordinator for all the COVID efforts in the UK amongst the different stakeholders and communities. They launched it 2 weeks ago. I can share with Graham the presentation and the WebEx. They did a session like us. And I don't know if this is going to be the coordinating body, but it seems to be the most structured one at the moment. It's very scientific, but also it helps business as well, which is why I was involved, yeah?
Okay. That'd be very useful, thank you. I think one of the things I would say is going back to the not-for-profit group that we've just gone about to set up. The reason we're doing this is that we went to some of those agencies and said, here's our ideas, this is what we want to do. Almost uniformly what we got back was, we're only interested in repurposing drugs because it's going to be quick and we want to do those. And frankly, we just found it too slow to deal with them. So we went off and raised our own cash. Said we got 100K in a day. And that's enough to get us to screen the library we have in mind. Now, obviously, when we find hits, because we think we will find hits at this library, it's been used before, other viruses and found hits, we'll then try and plug into the Wellcome Trust and the Gates and other groups. But currently, I have to confess, I'm finding them quite bureaucratic, even in these situations. It's a natural. I know. Why do I sound surprised? I was trying to be tactful and diplomatic, but it's a natural way of working. And so, unfortunately, you have to fill in the forms and have the talks, and they have to tick boxes, and it's just very difficult to operate. So we just said, "The hell with this! We'll go and get some money ourselves."
So, mindful of the time, folks.
Yeah, Tony, can I just interrupt quickly? We've got a hand up in the room. Okay, there's a hand up. Ahmed has got his hand up in the room.
Where's Ahmed?
Is Ahmed there?
It's Catherine.
Oh, Catherine. Oh,
where's Catherine?
Oh
Catherine, yeah?
Yeah.
Catherine.
Oh,
you haven't got.
She's not on.
Yeah. That's a disappointment.
Well,
Marty.
Anyway.
Yeah, I'm sorry. Yeah. I was very confused. So carry on, please.
Hi, Eddie.
Hi.
I just want to ask you about the death rate, and currently we think it's 0.5 to 1%. Actually could turn out to be significantly lower than that, or do you think, or maybe we don't know at all?
Okay, the honest answer is we don't know at all. But if you ask me to make shit up, basically there's two studies I'm using. One is the Korean study, which actually is great because they actually tested so many people. They picked up most of the asymptomatics and the low symptoms, and of course the very sick people. And there the death rate was 0.6%. The other one I'd look at was a study done on a much smaller group of people, which was on the cruise ship, where they had about 3,500. 700 became infected, diagnosed, and they really screened these people like crazy. And what they found was 18% were totally asymptomatic. They had no symptoms whatsoever, which is fascinating. And that's, it's a small group, but it actually gives you some numbers. So my guess is, based upon those two studies, we're going to find the death rate round about the same as flu, 0.5% to 1%. But again, the warning is it's going to vary enormously according to the culture you're in, the group of people you're in, how many people are old in that population, how they work together. We're going to find enormous differences.
Yeah, I mean, can I, Graham, I'm mindful of time here, but can I just ask, at this time in the show, we like to ask people from different parts of the world. So I'd be really interested, Vikas, can you tell us how it's going in India?
Hi, Tony. Hi, everyone. The lockdown continues here. I think we did this lockdown almost like a week ago, 2 more weeks to go here. And yeah, the death rate has not been that high. I think the last reported number is around 47 or 48. And roughly about 1,800 people have been infected so far, tested positive for this coronavirus. So I think what is happening here is we are not testing the entire population. we are testing people who have symptoms. And this is what is coming in the news also, that see, as a country, we have such a large population. We can't test everybody because we don't have the resources, and this situation has come all of a sudden. So maybe the country was not prepared, and that is why the easiest option for us was to go under lockdown, just stay indoors.
And is there social distancing? But having been to India several times, I know it's a complex country, for
right.
social distancing.
Yes, social distancing doesn't really fit into this country. You have personally experienced that.
Yes. It trains and buses. Yes.
I hear there's a few brutal beatings as well if you're not complying.
That's true. No, the administration has to be a little strict. Why? Because people, because we have less space, more population. So the concept of social distancing has come as a very new concept for the entire population here. People are learning, people are adapting. People who understand that it is a very severe thing and it can affect their health are staying indoors, not really venturing out. But then people who are not educated, people who take this virus as just another virus, they are into that adventurous mode. They want to go out, explore what is happening. And so I think we are learning, as a society, we are learning. But I think if this week, especially till the 10th of April, it's a very critical time for us, because the virus has gone into the third stage, the community infection, and which is what the government is trying to avoid. Because if it gets into the community here, it'll be a disaster. I mean, nothing can stop this virus, from taking many lives.
Oh, goodness. Well, Vickers, please, can you keep coming back, because we really want to keep hearing what's going on there.
Yeah.
I really appreciate
that. Sure.
Graham, how are we doing for time?
Good night. We're doing okay. I mean, I know we've got to wrap this up. There are a few people though I'd like to hear from, and one is Phil, because not only is he really patient and but he's got a really beautiful poster behind his head, and I want to take a photo of it once he's on speaker.
Graham, it's a green screen. I nearly put one on for Dave, but that's enough.
Okay. Wondering if you have a question.
No, I don't actually, because one or two of the questions I had have been covered really, really well. I'm coming at it much like I'm looking at Diane as well here, from a slightly different perspective, in that we're working for companies that are actually looking at different levels of approval and working through the trials that are currently ongoing. So hopefully I'll be able to share a bit more with that on that topic in the coming days and the coming weeks, as I go back to the ranch and try and get through a few confidentiality issues.
Right. How about we just put up hands? Yeah, Diane would like to speak just briefly. Sue and Nicola would.
Nicola's there. Yeah.
Oh.
Hello. How are you?
Great. How are you?
Very quick, because you said something, Eddie, that just sent my heart crossways, especially from a trial design and a trialist perspective. And I just wondered, you'd said that, initially you were dismayed at the quality of some of the data out there and the dreadful quality of some of the studies out there. And as someone that owns and runs a CRO, that bothers me considerably. So I suppose if you had to even give a couple of solutions, even one solution, how do we address that? And is there a way to have a more collaborative, informed approach in terms of how we're designing studies? Because to me, that's a critical part of getting answers to all of this stuff. Does that make sense?
Yeah, I'm going to be even more depressing. So if you look at respiratory, I've got to go back to my RSV story again. You use small molecules. Gilead have an inhibitor of RSV called prestatavir. It went into a clinical study in bone marrow transplant patients. The patients who entered with lower respiratory tract infection, that inhibitor did absolutely nothing. The patients entered with upper respiratory tract infection, and you measured the progression to lower, it almost got a p-value, right? But if you subset analysis with people who had lymphopenia, it worked brilliantly. So the lesson here is, for me, if you take a respiratory disease and people have established late-stage pneumonia and the effects that you're seeing with COVID, it may be very hard to show any compound actually has any effect, because to reflect back to the question earlier on, a lot of the disease is going to be driven by immune pathology. Virus may not be the major driver there. So again, I personally would select a population. I'd select people over the age of 70 who had comorbidities, who are definitely positive, but early in their disease. And that's the group I would do a clinical study against and see whether or not you can stop them progressing to lower respiratory tract infection. My concern is good drugs may go in poor studies, not be shown to be active, and we just will let things go. By the way, if you want to read an appalling paper, read that French study. It is the worst study I've ever read.
I don't know, I feel like I could give it a run for its money, but...
Oh, okay.
Graham, I think
no, Tony, can we just, because there was a hand from Suzanne and Diane as well, so if we could have Diane ask a question, and then Sue, that would be great.
Yeah, I mean, hi Eddie, I don't think we've ever met before, but I have worked in regulatory affairs for a very long time, and so my question was, you know, you've set up this group to look at things, and have you thought about, you know, interacting with regulators, innovation task force, that kind of thing, to make sure that when we do have to mobilize to get the next treatments for the next COVID pandemic, that things are going to be ready and, you know, meeting the regulatory requirements, because obviously we mentioned bureaucracy before, and obviously this is a bureaucratic thing that you need to get past in order to treat patients. So
it's bureaucratic, but I think it's essential. And I think
absolutely,
these small studies disregard regulations to their peril. So here's the thing, here's a confession. What people like me are good at is the early stuff, taking them through development up to clinical studies. And we've got regulator advisors and things. We've sort of seen it from afar, but our model is that's not what this not-for-profit group will be doing. We will take it up to the point of clinical studies, and then we will seek a partner. It's almost certainly going to be a big pharma partner who have the infrastructure and the machine to take things through. And that sounds like I'm abdicating responsibility. I'm not. And I know that you have to be aware of regulatory constraints very early on in the process, or else you can develop something that's not going to go forward. But we're going to basically take it up to candidate selection and then through the IND enabling studies, and that's where we stop, because we have no expertise and no knowledge in that area.
Okay, and then finally Sue,
Finally, where's Sue? Our Sue, where's Sue?
Here, hi.
Hi, Suze.
Hi.
How are you doing?
Yeah, good. So, as you know, Eddie, my heart is with small molecule virology, but now I have to talk about money and venture capital. Do you think the venture capital world is irrelevant in this scenario? So,
no, I don't. And we've had a big squabble within the group setting up this not-for-profit group about it. I think, so first of all, I think we should look, once we have some hits, we should go to a number of VCs, but we have to be clear, they're probably going to do this without making any money.
Yes.
Now, we have to be clear, because I think we shouldn't paint VCs as some horrible beast that has no humanity. They want to contribute to this. You're obviously one of those people. We obviously, they want to contribute to this as much as anyone else, as long as it's absolutely transparent. There are mechanisms by which you could make a not-for-profit making organization have a profitable arm, by the way, but we're not going to do that. We just want to make this absolutely transparent. But we will be calling upon some investors when we have hits, we've got something tangible that makes sense, to say, right now we need 20 million to take this molecule forward until IND enabling. By the way, our ideas extend out to Ebola and other viruses as well. But we start from coronaviruses. That's where the issue is today.
So that was a beautiful segue to finish on, Sue, because our next series is going to be on financial impact and raising money in this environment, and how do you find pharma partners that may be distracted. So Graham, do you want to say any more about that, my friend?
Well, you know, I just want to squeeze one or two more people in, to be honest, before we end. I mean, I know we're all pushed, but Joanne knows Eddie, and I'm sure Joanne or Sue Reese. I think Joanne. Would you like to ask something quickly?
Okay, very quickly. Hi, Eddie.
Hi, Joanne. Long time no see.
Yeah, absolutely, absolutely. Quick question. You alluded to earlier that people generally are thinking that a repurposed drug is going to be the quickest way to get a therapeutic against this particular virus. Are any of the ones we talked about even likely to work? I mean, I know famous scientist Donald Trump said it's a game changer, and everyone started drinking fish tank sterilizing fluid.
Yeah. I think the molecule that I'm not a big fan of repurposing. Because when you developed a compound, you're normally targeting something very selectively into one target, and it's hard to get breadth. The one I'm more interested in is a Gilead compound, the nucleoside analog. I think that could show promise. I'm a little bit cautious about the toxicology behind that molecule. The one I personally don't favor is the fish tank molecule. I don't see how it's working, and that compound's been dragged out the cupboards for HIV, hepatitis C, Ebola, and SARS every time that comes out.
But the interesting thing, Eddie, is that hydroxychloroquine is also an anti-arthritic.
It is an anti-lupus.
Yeah, so, there's a very intriguing connection in the end between IL-6, I heard salicylic acid today, so there's something about inflammation at the end, which is actually what we need, that support there.
Well, that's why I would support Tony doing a clinical study, a proper controlled clinical study with measurable, relevant clinical endpoints. What I would say, however, is currently we're putting hundreds of thousands of patients on a compound, and there's a lack of supply for the people who have lupus and arthritis. And I think people, they're not trivial diseases. So we just need to be careful that we're not knee-jerking. Proper clinically relevant studies need to be done.
Thanks. Now,
we've got one more burning question from Victor. One more burning question from Victor. Can you put your microphone on, Victor? Yeah, you got to go. So it's usually the bottom left hand, or that's it.
Hi there, Eddie. We
Hello.
met once at the BOVEC meeting. I have a question for you. Coming from, we're working on diagnostics and self-monitoring of respiratory conditions in the home to prevent people having asthma attacks and exacerbations. So one question I have for you, is there any way in terms of the mechanism of the transmission and replication of the virus, you could actually identify people that have had the upper respiratory infection before they get the serious lower respiratory infection? Is there any way, do you think, you could identify people that have susceptibility or immune responses where...
that's interesting. Yeah, that's an interesting question. I think the group that I would look at is a different, that's not a question in a different way. I would look for the comorbidities, people of high blood pressure, cardiovascular, and say, if we can stop them as early as we can. Again, I'm going to be very unscientific. I'm a little bit nervous about some of the people who are on ACE2 inhibitors, because the evidence is that induces higher concentrations of ACE2 on cell surfaces. And I think there's something there that we need to try and investigate more over time, is when we look at the epidemiology, was there any link for people on ACE2 inhibitors? If you're a diabetic like myself, there's a compound called pyoglycoside, which also inhibits ACE2 inhibitors. And I can tell you, I took myself off that about a week ago. Now that's a non-scientist. I've been saying about how good we should be scientifically, a lot of non-science in there. In terms of recognizing people of upper respiratory tract, as far as I know, the only way to do that is to do the scanning, the x-rays, which are very distinctive for COVID. They can really diagnose it very easily with the x-ray shots of the lungs.
Graham, have we finished squeezing now?
Yes, we've finished squeezing now. I just, I'd like to say, thanks very much to everybody. We are looking forward every Tuesday and Thursday at 2:30 p.m. to continue this, coronavirus discussions. We want to hear from everybody, their angles really on, how their businesses are dealing with this, and also their strategies. So we particularly want to hear from investors, potentially pharma companies, Cateray would be very interested to get any insight you have, and you know, other areas. You know, how's this going to impact, just so that we can give everybody insight into how they should be changing their business thinking and strategies. So I'll leave it there, Tony. Thank you very much.
Folks, can we just say, Fab, this is meant to also be a self sort of help group, and we will find time to keep each other buoyant, because seeing all your smiling faces certainly, even though I didn't see Gaz's shirt, which I think he's hiding somewhere, but he's always
got a fish there instead. A
feature of this. And be careful out there, and we'll meet again next Tuesday. And thanks, and bye for now. Ciao.
Thanks, Eddie.
Thanks, Eddie.
Thank you. Thanks, everybody.
Bye for now. See you. Bye.
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 Bye. Bye. Yeah.
Will.
Eddie.
You're very quiet.
Eddie.